did a couple of weeks ago, and then I was in trouble. So right now for the extra credit, because of my poor skills working with Canvas, I couldn't quite figure out how to give you these points as extra credit. So they actually, right now, they're considered as part of the grade. And if you didn't do so well on this, it actually lowered your grade. So this is something that I will have to resolve, which is going to be my homework for today or tomorrow morning. So I will try to figure it out how to actually turn these points into extra credit. There's some special ways. The problem is that I know how to do it if this was an assignment, but this is a quiz, and I didn't see an option of giving you a quiz as an extra credit. But I will keep looking, and hopefully I'll find something. All right, so what's today? Today is 23rd, Monday, 11 o'clock. We have a Thanksgiving break coming up. And then, so the next lecture after this one will be a week from today. And that's going to be the last lecture, right? And then you have a test Wednesday of that week. And then basically we're done. I will give you a review session for the final, give you some pages to read in the textbook so that you don't have to read the whole thing, but just focus on the most important aspects.
Do you know how many questions are on the final or how long it's going to be yet?
Yeah, normally the final is 60 to 70 questions. So I'll keep it at that. Depending on the difficulty, I know you probably liked easier questions, but more of those, right? So that you can actually, if you miss one, you can get more points on the other one, on the remaining ones. Yeah, so 60 to 70.
I was more like thinking about calculations and how long those might take me.
Yeah, so as far as calculations, so this is not a math class, right? So as you already noticed, I don't really give you as far as calculations, most calculations that I present you with are those that you can do without a calculator, right? So you still like to use the calculator, but yeah, so there'll be if there is math, it will be pretty straightforward math. Okay, so
thank you.
Nothing challenging. Any other questions? So we gonna start chapter 12 today. Chapter 12, and so we have 2 lectures on chapter 12. So this material is pretty much straightforward. It's mostly descriptive. It's what it's how many biologists do their work. They basically, they treat complex biological systems with how different enzymes or proteins interact with each other, how the signal is transmitted from the outside into the nucleus, and so on and so forth. So this will be more sort of a biological presentation, not so much biochemical, right? So we're not going to be talking too much about chemical bonds or hydrophobic or hydrophilic effect. It's more about which protein interact with which enzymes and how the signal is transmitted from one molecule to another. So, all right, so let me share my screen. Hold on a second. So I didn't forget to record, which is good. I'm recording this. All right, before we begin, any other questions? All right, so biosignaling. Now, so what we're going to focus on are general features of signal transduction. right? So some general pathways which are utilized in signal transduction. As I mentioned to you many times, biology is highly conservative, right? So you will see the same principles, just in different context, right? So nature likes to utilize same things over and over. Otherwise, there will be total chaos. It's just some slight modification of one method that can be applied to one signal transduction pathway or another, but the principles would be the same behind both. So we'll start by discussing G protein coupled receptors. These are some of the most common ones. We'll talk about enzyme linked receptors, ion channels, right? So those will be gated, right? So for example, you can have a ligand that binds to ion channels and opens the ion channels or closes them. And finally, we'll talk about physiological processes which underline the signal transduction. So let me, all right. So obviously in a multicellular organisms, cells do not live by themselves. They talk to each other. They have to talk to each other, right? They need to know how much they need to grow, right? So we don't want to have tissues outgrown because the cells continue to grow and don't know where to stop. Some cells are even told to die, right? So we talked about apoptosis, and we'll talk more about apoptosis, programmed cell death. When an organism basically is telling the cell, you need to die, because otherwise, if you don't die and continue to divide, there'll be cancer, right? And so this cell totally voluntarily will undergo apoptosis through what's known as a death signaling pathway, through death receptor. So some examples of such signals, so antigens, hormones, neurotransmitters. We'll talk about light, perception of light, touch, pheromones, signals that cause changes in cells composition and function, differentiation. So again, also an important aspect that that cells will have to form a different tissue from the original tissue. Otherwise, this original tissue will outgrow all the other tissues and lead to uncontrolled proliferation and cell death. Growth and size, sexual versus asexual cell division. So we'll touch on these aspects of signal transduction. So in this chapter, we'll focus mostly on signal transduction through receptors, right? So receptors is a membrane bound or could be soluble protein complex that exerts some kind of physiological effect. So we call it intrinsic effect. And that will be after binding of its natural ligand. So for example, with G protein coupled receptors, we'll talk about epinephrine receptor. So epinephrine receptor, you know that these are very important receptors in situations when you need to mobilize your energy and escape danger, for example. So these are fight or flight receptors, right? So for example, when the blood flow will be diverted from the stomach, which toward muscles, for example, or the heart, right? So for example, if you're presented with danger and you need to run away from it, digesting food is the last thing you need to be thinking about, right? You need to run, you need to run. So, but after you finish running, then the epinephrine receptor has to or the signaling through epinephrine receptor has to be shut off, and the blood flow has to go back to food digestion. So epinephrine receptor is a great one from that perspective. We'll talk about enzyme linked receptors. So these are insulin receptors. In what we will see in signal transduction, that one of the main type of reaction which is involved in signal transduction is phosphorylation. And can you think of amino acids that can undergo phosphorylation? Just give you a hint, they have hydroxyl groups in them. So what amino acids have hydroxyl groups?
Serine.
Serine, that's one, yeah. There's another one.
Tyrosine.
Tyrosine, perfect. Right, so those are the ones that you mentioned are important from the phosphorylation perspective, right? Just think about it. So if you have, let me just draw this. So let's say you have tyrosine, right? You have tyrosine. We'll have an OH. And do you remember the name of enzymes that phosphorylate hydroxy groups? What kind of enzymes phosphorylate hydroxy groups? Remember, there are two kinds.
Is it the ATPases?
ATPases. So those actually hydrolyze ATP to ADP and inorganic phosphate. So these are different. Starts with a K. What do I write here?
Kinase.
Kinases. Perfect. Kinases. And so now look at the difference in structure. It's huge, right? So here, even though both of these are polar, but this is neutral, it's uncharged. But when you have a phosphate, now you have two negative charges, right? And so there are lots of enzymes that may bind electrostatically to this phosphate, right? So we'll talk about specific domains on these enzymes that recognize phosphatyrosines. And by doing so, then you can recruit this enzyme, for example, to this site. And this recruitment will result in signal transduction. And so these kind of receptors are very important. Now, ligand-gated ion channels, obviously, whenever you have an ion channel in the cell membrane, we have to have the ability to open that and to close that. Otherwise, that would result in depolarization or hyperpolarization of cell membrane. So, and in many cases, ligands will serve such a role. And for example, acetylcholine, which is a neurotransmitter, right, could be one of such ligands. Let's see. In fact, acetylcholine receptor, it's a non-selective. There are lots of selective. We talked about ion channels, and we even discussed how nature created selective ion channels. But it turns out that with acetylcholine receptor it's actually a non-selective receptor. So it can allow the passage of sodium, potassium, and even calcium through it. Integrin receptors. So those are receptors which mediate or facilitate the cell extracellular matrix interaction, ECM. Right, so the cell has to be attached to something, otherwise it will just float, go away, get in the bloodstream, and just get washed away somewhere else. So it has to be attached to its to the tissue where it's performing its function. So extracellular matrix. But the function of integrin is not just attachment, even though that's kind of the primary one. But there are obviously these receptors. Whatever happens outside of the cell gets transmitted inside the cell. And so the integrins are kind of type of sensors, which are telling the cell, okay, so the extracellular matrix, there's something's going on there, and it's time for us to divide, right? So for example, it can also organize cytoskeleton so the cell can change shape, right? All right, so we'll talk more about that later. And nuclear receptors. Now, do you guys remember what? Okay, that's already written here. So steroid receptors. So obviously sterols, right? So steroids, compounds derived from cholesterol. Many of them are hormones. Basically, they regulate the expression of genes. And the genes will control development, homeostasis, metabolism, And so, so these receptors, steroid receptors, directly bind to DNA, and regulate the expression of adjacent genes. So let me quickly write this down.
So would that be like hormones as well?
Yeah. So steroidal hormones, that's right. Expression of genes. And so obviously keep in mind that steroids by themselves, they're hydrophobic, right? So it's very easy for them to actually cross all sorts of membranes. They can cross the cell membrane, get in the cytoplasm, they can cross the nuclear membrane, get towards the DNA, and bind to the expression factors or transcription factors. Transcription factors, which are proteins which regulate the expression of genes in the DNA. All right, so this is what we're going to talk about. So the 5 types of receptors. Let's move on. So here we have specific ligands. Now this is not quite different. So you saw insulin, ferric iron. So we know that iron is extremely important for the survival of organisms. We even know, we talked about even heme, right? Heme that in our bodies that is important for the transport of oxygen. But also iron plays a big role in enzymatic reactions. And so remember I told you one of the main mechanisms for bacteria to acquire iron is through the mechanisms involving molecules known as siderophores. Do you guys remember what that was?
I don't remember.
Anybody else remembers? No? Siderophores. So right, so these are peptides in most cases, which bacteria produce and release in the extracellular environment. And these peptides will bind iron and bring the iron back inside the bacterial cell, release the iron, and then go out again, hunting basically for iron. But also bacteria have on the outer in the outer lipid layer, it has lipoproteins, which actually can capture ferric ions from the environment. So we talked about epinephrine. Now this one is an interesting one. Vascular endothelial growth factor receptor, okay, VEGF, so-called VEGF. Very important from the perspective of drug design. What do you think this receptor does? Or what do you think this factor does that binds to the receptor? Just look at the name of the receptor. You know vascular, you know endothelial cells, right? The cells which line the inner surface of the blood vessels. Growth factor. So what do you think this receptor is responsible for?
Oh, sorry, growth.
Growth of what?
I guess your body. I'm not sure.
Well, growth of what's reported?
Okay, vascular endothelial.
Endothelial cells. So there is a process known as angiogenesis. Angiogenesis. Now there are obviously the cells can grow and the new that's a the growth of blood vessels is a common process and physiologically necessary. But there is whenever a patient has a tumor. Now, for example, a tumor, which is let's say this is a blood vessel, right? The blood vessel, and the tumor starts to grow near the blood vessel, okay? And so after a certain time period, the tumor becomes huge, and there is still this blood vessel, right? And that's the tumor. So, so the blood vessel will deliver oxygen, will deliver nutrients. So the tumor cells, which are on the outside of the, which are close to the blood vessel, they'll be happy because they're gaining lots of oxygen, they're gaining lots of nutrients. What about the cells inside? Will they get oxygen? Will they get nutrients? Obviously very little, right? And so what tumors do, they basically secrete various growth factors, which will lead to the process of angiogenesis. So in other words, the growth of blood vessels, which will provide the tumor cells inside with oxygen and nutrients. And so what they will do, this will not be, let's say, I'm gonna draw another tumor cell like so, right? And so let's say this is the, hold on a second. So some of this tumor will, some of this blood vessel will branch off and go through the middle of this tumor, for example, right? So the oxygen and nutrients, so let's say this is the blood. So the oxygen and the nutrients can be delivered to the cells which are inside the tumor, right? So so vascular endothelial growth factor is one of those receptors which the tumors will make sure that these blood vessels express in large quantities, right? So they can be utilized for growth of the blood vessels. And so it's not surprising that actually vascular endothelial growth factor itself and its receptor are huge targets for cancer drug design. And a number of drugs actually have already been approved and used in the clinic to treat patients with solid tumors. And one of them is, for example, it's an antibody Avastin. It's used to treat, for example, colon cancer, even glioblastoma, brain cancer I was telling you about, which to me is still a kind of a puzzle. I still don't quite understand how an antibody can cross the blood-brain barrier. Remember I was telling you about BBB, the blood-brain barrier? But Avastin is still moderately effective against glioblastoma, which is a brain cancer. Basically inhibiting angiogenesis, the growth of the blood vessels, which will provide tumors with oxygen and nutrients. Okay, any questions? Sorry, it took a bit longer than I expected, but let's move on. All right, so there are 5 features that we can identify in signal transducing systems. So one is specificity, right? So here, we want to have a receptor that actually will have a binding site which will specifically recognize its ligand, but not other ligands which may be structurally similar, right? So that, remember, the what we're looking for is a low KD. You guys remember what KD stands for? What's KD? K is a constant. What's D? Dissociation. So this is dissociation constant. And remember what number we're looking for? So most drugs work in what range of KD values? So there is a molar, right? Millimolar, micromolar, nanomolar. What are we looking for? Picomolar? No, don't remember?
Millimolar.
Millimolar, perhaps, but that type of interaction will be quite weak, right? Which will allow for other structurally similar molecules to also interact with the receptor. And many of them may even form tighter complexes, in which case they will block the receptor. We don't want the receptor to be blocked, right? So how about nanomolar? That's a good number. That's a good unit. Nanomolar. So in fact, so we have millimolar, micromolar, nanomolar. Remember, the nanomolar is 10 to the negative what? 10 to the negative
9?
9.
It's 10 to the negative 9.
9, perfect.
So what about micromolar? Could you use that?
Yeah, so yeah, there are many drugs. So there are obviously many exceptions to this, and there are many drugs in the market which let's say antagonists, and they block receptors in the micromolar region, and many of them are successful. But obviously, the smaller this number, the more specific your interaction will be. So that's what you're aiming for. And in fact, many times when from my own research, when my group would submit a research paper for publication, for example, to Journal of Medicinal Chemistry, which is considered one of the higher ranked journals in medicinal chemistry, and my compounds, even though mode of action is very interesting, the assays were well done, and the target is very interesting, promising therapy that could arise from that. But the compounds are micromolar, and oftentimes such manuscripts would be returned to me, and the excuse that the editors would use is that, oh, your compounds are micromolar. And so I would even give examples. Look, these other drugs on the market, look at cisplatin, a cancer drug. It's micromolar. It kills cancer cells in the region of 20 to 25 micromolar. And yet it's used to treat so many different kinds of cancer, so successful. And yet the editors are still looking for small nanomolar compounds. All right. So this is something to keep in mind, specificity. Now, amplification. So we will see with GPCRs, especially G protein coupled receptors. The concept is very, very, very well illustrated by GPCRs, where basically from a single molecule of a signal, it gives rise to multiple outputs. Modularity. So here is receptors have a way to build signal transduction pathways in such a way that they can be multivalent, right? So, for example, one player in the signal transduction pathway can branch off and have another function attached, another function, and can branch off and initiate a signal in this direction, that direction. And phosphorylation plays a huge role in that. Remember, I told you that you can have multiple tyrosine residues in different parts of the protein, and phosphorylation of one of them can lead to one pathway for signal transduction. Phosphorylation of another one can lead to another. So so receptors have multivalent affinities, so very important. Now, desensitization, adaptation. So we'll talk about that in some detail. So when the signal is persistent, right? So the receptor finally gets tired and says, look, this is enough. Enough of you, I've seen enough of you. Let me make my response to you weaker, right? And so there is a pathway for the receptor to be desensitized or become adapted to the signal. And you oftentimes will see that if you take a medication, originally you take a small dose and it helps you, but then you find that you need larger and larger doses of the same medication to produce the same kind of effect on you. Integration, right? So integration. So we see that in the nerve system, where basically there's one particular cell that can actually receive inputs from other cells, and it will basically sum up all these inputs and produce one response based on the summary or the sum of all these inputs, right? So it could be concentration, could be voltage. And finally, localized response. So what this has to do is that it's a way for us to not to transmit the signal to large distances, but rather to have a message remover where the message will only be transmitted to in the local area, rather than distant sites in a cell or in the body. So this is something which is known as a localized response. All right, any questions about this slide?
So all of this signal transducing, this whole system, is this happening inside the cell, or this is just what happens throughout the body? Are these all inside the cell?
Well, so yeah, so we're mostly talking about receptors in the cell membrane here, right? So everything we'll see will be actually the cellular responses to the changes in the extracellular environment. Yes, correct.
Okay.
Yeah, so this is just a slide that I was telling you about. So what's the units for this? So kg 10 to the negative 7th. Can you convert 10 to the negative 7? So this is molar. Can you convert 10 to the negative 7 molar into micromolar or nanomolar, for example? So how many nanomolar? What's the concentration of this expressed as nanomolar?
Would it be 10 to the negative 2? Or am I going the other way?
Oh, just get rid of the log. So so micromolar. So just keep in mind. So 1.0 micromolar is 10 to the negative 6 molar. So how many micromolar is 10 to the negative 7th? So reduce this by another order of magnitude. So 1 divided by 10, what's that?
Just 0.1?
0.1, yeah. So 0.1 micromolar is the concentration, which could be a reasonable KD. You can also express this as 100 nanomolar, right? 100 nanomolar. All right. So this is just a cartoon-based representations of what we talked about. So G protein coupled receptors. So here is the ligand. So there's a receptor coupled to G protein. So ligand comes in, binds to the receptor, which releases G protein. Now we'll see that actually it releases some of the parts of the G protein. And this G protein then binds to an enzyme and releases, and the enzyme will produce another molecule which will migrate into the cell. So oftentimes we refer to this as second messenger. Second messenger. Right, so if the ligand was the first messenger, this will be the second messenger. This goes into this one goes into the cell and then transmits the signal wherever it's required. Now here is the receptor tyrosine kinase, for example. So we talked about tyrosine. You can see here it's possible that, so this is the extracellular domain, ligand binds to it. The two arms, which are intracellular domain of the receptor, can come close to each other, and it's often possible that they each phosphorylate each other. So in other words, both of them have tyrosine residues and enzymatic activity. So this phosphorylates that one, and this phosphorylates that one. So it's something which is called autophosphorylation. Kinase cascade, and then goes inside the nucleus and results in the expression of a particular protein. Now, nucleoreceptor, this is what I was telling you about, right? They just basically sterols or steroids basically cross the cell membrane, nuclear membrane, and binds to a transcription factor, which initiates the synthesis of messenger RNA, and then a protein will be synthesized from that. Now, here's the ligand binds to an ion channel, which by itself is closed. When the ligand binds to it, the ion is allowed to move down its concentration gradient. So this is gated ion channel. So this is just a cartoon representation of what we talked about. All right, so let's talk more about G protein coupled receptor signaling. Let's see. Yeah, let me tell you here maybe at this point. So this data may be already outdated, but there are 800 genes encoding GPCRs. Extremely large diversity of these kind of receptors. These GPCRs are overexpressed in 20% of cancers. And part of the reason for that is because many of them are responsive to growth factors, right? So cancers want growth factors. They want to divide. And growth factors help them do that. So 1/3 of drugs target GPCRs. And it's also been estimated that 100 GPCRs, 100 GPCRs are what's known as orphan. Does anybody know what orphan receptors are? So orphan receptors are those whose ligands we don't know. So we found the receptor, but we don't know what the ligand is and what the response would be when the ligand binds to its daddy, right? Orphan. So orphan is like orphan receptor is like a known, like the children don't know their parents, right? So so G protein are heterotrimeric, alpha, beta, and gamma. They are membrane associated, and they bind GTP. So one of the prototypical G protein is RAS. So here's a crystal structure here. Just for us, this is just a X-ray structure. Now, keep in mind that many of these are the problem with GPCR research is that they often hard to crystallize. They often hard to crystallize and obtain X-ray structure. So this X-ray was obtained with magnesium in it, and there is inside there is a molecule of GTP, but not exactly GTP. It's a molecule of, let me actually write it down. What we have inside is ribose, G, phosphate, and then NH and phosphate. I will just neglect all the other oxygens. So why do you think we have a nitrogen here, not oxygen? So normally, if you want the obtain a crystal structure of biologically significant or the biological equivalent of RAS protein, you would crystallize RAS together with GTP. But what's the danger here? Remember, RAS is a GTPase. If it's a GTPase, what does it do? What do GTPases do?
They phosphorylate.
They would do what?
They remove a phosphate.
They remove the phosphate. So GTP will become GDP. And remember, so I'll draw the reaction for you actually. I'll draw the reaction for you. So you have G, oxygen, phosphate, oxygen, phosphate, oxygen, phosphate, GTPase, right? GTPase. G, oxygen, phosphorus, oxygen, phosphorus, oxygen. So this is diphosphate, this is triphosphate, right? So this is a GTP, this is GDP. So why did researchers not use GTP to obtain the crystal of this, but rather use this molecule, which looks similar to GTP, but instead of oxygen, it has a nitrogen. Can the intrinsic GTPase activity break down this molecule? Basically, what we will need to do, we need to break this bond. So let's make this slightly bigger. We have oxygen, nitrogen, O minus, O minus. So what is this bond now? PN. So if this is here, phosphor monoester, right, bond? What is this bond here? Phosphor what? Phosphor monoester. Let's look at this. Then what is this? Well, just think of carboxylic acids. Replace oxygen by nitrogen. What do you get? What's the difference between this and that?
Is that amine?
No, it's not an amine.
It's an amide.
Amide. So what can you say about hydrolysis of, and what's this one?
And ester.
Ester. What can you say about the relative stability of these and relative susceptibility to hydrolysis?
The amide is going to be less susceptible to hydrolysis, and it's more stable.
And what do you say? What can you use the same reasoning here?
Yes.
Yes. So now this is phosphoramide. This is phosphoramide, right? So phosphoramide is going to be much more stable than phosphor ester. So this is the idea. This is why the researchers used the equivalent of GTP, which is oftentimes referred to as non-hydrolyzable. Non-hydrolyzable. And it's commonly used in research. Because it behaves similarly to GTP, but it's not hydrolyzable. And you can form stable complexes. Okay, does it make sense? All right, so what I'll do, I know you're taking notes, but I will also upload this discussion on Canvas so that you can go over it in your free time and again think about esters versus amides. All right, so epinephrine. Epinephrine interacts with the cell via GPCR. Adrenal glands, so you know that that's where it's made, right? Just above the kidneys, mediates cell response, mobilization of energy. I was telling you. Binds to receptors in muscle, liver cells, inducing breakdown of glycogen, right? So there lots of glucose goes into the bloodstream when we need to, when we present it with a fight or flight situation. In adipose tissue, right, adipocyte cells, induces lipid hydrolysis, right? Again, to generate energy. And heart rate in the heart increases heart rate. And so you can see actually here the natural ligand epinephrine binds with a KD of 5 micromolar. Now this is agonist. Remember, agonist versus antagonist. I don't think we specifically define these, but just based on this, just think about it. What's the difference between agonist and antagonist? I think the term kind of speaks for itself. What does the agonist do compared with the natural ligand? And what does the antagonist do compared with the natural ligand?
Agonist makes like a physical response, like a physiological response.
Yeah, perfect.
When it binds to its receptor.
Yeah, so it mimics the natural ligand, right? An antagonist.
Oh, sorry, it does the opposite.
Does the opposite, right? And in general, what you may notice is antagonists in general have larger structures. Like if somebody showed this to you, right, and it looks very similar, you may say, oh, this is agonist as well. But if somebody shows you propranolol, for example, and you can see this huge naphthalene ring, this is a very good indication that this molecule is actually an antagonist. It basically binds to the entire pocket and can totally blocks it like a lid, totally blocks the binding site, and you can see very low KD value, right? So this is 4.6 nanomolar, 4.6 nanomolar. Propranolol is an excellent old drug for because it blocks the beta. Because it's a beta blocker, blocks beta adrenergic receptors. All right. So now let's look at in some more detail as far as what happens with the GPCR once the signal is transmitted into the cell. So how does the GPCR work? Let's look at more detail. So epinephrine binds to the receptor, which is a transmembrane, right? It's a 1, 2, 3, 4, 5, 6, 7. So it has 7 transmembrane domains, right? And how do we know it has 7 transmembrane domains if we don't have X-ray structure? Or not even with X-ray structure. How do we know that it crosses the cell membrane 7 times? Remember the plot that one can construct?
Using the polarity of amino acids in its sequence?
Polarity in its sequence, and you plot that somehow, remember?
Yes.
Do you remember the name of the plot?
I remember I just did it on the homework last night. I don't remember the name of the plot though.
Looks something like this. What's on the y-axis? Anybody remembers?
Is it the hydrophilicity?
Yeah, well, close. Hydropathy. Hydropathy. Right. So with this, if you see that there are 7 bumps here, well, this 7 bumps here, then it crosses the cell membrane 7 times. Hydropathy plots. Okay. All right. So here's our GPCR. Okay. This is the receptor. So it's a heterotrimer. Originally it's bound to GDP, right? So it's alpha, beta, and gamma. Now there is GS, it's called stimulatory. Now there's GQ, there's GI, there are different kinds of G proteins. So we're going to focus on GS in this class. So here there are three domains, right? Once the epinephrine binds to the receptor, the receptor undergoes conformational change, which is transmitted into the G protein. GDP leaves, GTP takes its position. This translocates along the cell membrane and binds and activates an enzyme, adenylyl cyclase. Adenylyl cyclase, which then takes the ATP, we'll see the chemistry on the next slide, takes the ATP, kicks out diphosphate or pyrophosphate, and produces cyclic AMP, cyclic adenosine monophosphate. Now, this is our second messenger. This is our second messenger that then binds to an enzyme known as protein kinase A. Protein kinase A that then results in phosphorylation of multiple proteins and enzymes leading to the cellular response. Now you can see here cyclic AMP is then degraded, reversing the activation of pKa. So here is the actual chemistry of what's happening here. So cyclic AMP is a second messenger. It's here, right? Second messenger. And so this is ATP, right? There are 3 phosphates. There's a triphosphate. And you can see here what will happen. So the way to really visualize this is this hydroxyl group will attack the alpha phosphorus. So this is alpha, beta, gamma, the alpha phosphorus, right? And the diphosphate will be a leaving group. Remember, this is a great leaving group, right? And you form a ring. You form a ring, cyclic adenosine monophosphate. And then when the signal needs to be shut off, this will undergo hydrolysis, and the enzyme which will do that is phosphodiesterase. Phosphodiesterase. All right. So let's look at the signal amplification. So I told you that with GPCRs, this is actually quite characteristic in terms of signal amplification. So here we have a few GPCRs lead to activation of few adenylyl cyclase. Every adenylyl cyclase then makes several cyclic AMPs. Each of those will activate several pKas. Each pKa will then phosphorylate thousands of glycogen degrading enzymes in the liver. And in the end, this will result in tens of thousands of glucose molecules released in the bloodstream. So here is some approximation of this process, right? So epinephrine, 1 molecule, epinephrine will bind to the GPCR. Alpha subunit, so 1 molecule, right? Alpha subunit will be produced, but then the epinephrine can actually dissociate and bind to another receptor. And so overall, 1 epinephrine can actually give you 10 alpha subunits, 10 alpha subunits, which will bind to adenylyl cyclase. Each of these will bind to adenylyl cyclase. And in this process will result in 200 molecules of cyclic AMP. 200 molecules of cyclic AMP then will bind to pKa and will give us 100 molecules of active. Per each of these molecule, it will give us 100 molecules of active pKa. Then each active pKa will give rise to 1,000 molecules of active phosphorylase B kinase. Now that's in the liver already. So inactive glycogen phosphorylase B will become active glycogen. So again, this is all phosphorylation cascade. Each of these will give us 10,000 of this, and so on and so forth. And so glycogen eventually will give us glucose 1-phosphate, and total 100,000 molecules of glucose will be produced from 1 molecule of epinephrine. So this is what's referred to as signal amplification. Now here's just a list for just a tabulation. Obviously, I don't want you to memorize any of this, but just to give you an idea of how many processes, as I mentioned to you, biology is a conservative area. And so this is how many processes are relying on cyclic AMP phosphorylation. And you can see here that pKa phosphorylates serine residues in most of these, right? Serine. And there's one threonine here in the protein phosphatase 1 inhibitor 1. So it's a serine threonine phosphorylation, which is performed by pKa, protein kinase A. Okay, so self-inactivation. Obviously, epinephrine signal is meant to be short-acting, right? And at some point we have to stop this, otherwise our heartbeat will go through the roof, right? And we'll not be able to handle the effect of epinephrine. So there are ways for us to actually to stop the signal. And so one of them is, for example, we can downregulate cyclic AMP by hydrolysis of GGP in the alpha subunit of G protein. Another way is through desensitization adaptation. Here, one of the molecules can come back, bind to the receptor, and basically tell the receptor, or reduce, for example, the affinity of the external ligand toward the receptor, and so the response will be weakened. So there are many ways to do that. And so this one is based on the hydrolysis of GGP. And this is based on the intrinsic GGPase activity of the alpha subunit, right? So the first couple steps are the same. Ligand arrives, GGP leaves, it is replaced by GTP, and then the signal will be shut off by hydrolysis of GTP to GDP. And then once it has GDP, it goes back, reassociates with the beta and gamma subunits, and in the process of waiting for the next ligand to arrive. Desensitization of adrenergic receptor occurs through actually storage of the receptor. So obviously, one way would be to simply take this receptor into lysosome and chop it up and get rid of it, and then resynthesize the new one when necessary. But obviously that will be a huge waste, right? So we already made it once. Why do we need to destroy it and remake it? And so cells actually found a way to store this receptor. And so the way this is achieved is once the alpha subunit has left, beta and gamma remain. And so beta and gamma, they're still attached to the, or undergo reattachment to the receptor. And that reattachment recruits beta androgen receptor kinase, ARK. So this kinase phosphorylates serine residues on the carboxyl terminus of the receptor, right? Creating the binding site for a protein known as beta-arrestin. Beta-arrestin then recognizes these phosphates and brings the receptor, causes the receptor to undergo endocytosis. Remember what is endocytosis? What does it mean?
It moves into the cell.
Yes. As opposed to when it moves outside of the cell, what is that called?
Exocytosis.
Exocytosis, good. So undergoes endocytosis into this vesicle, and so this vesicle has a bilipid membrane, lipid bilayer, and so the receptor will just rest there, wait until it's required, and then again once it's required, it will leave this vesicle, migrate back into the cell membrane, and will be ready for the reception of the next molecule of epinephrine. All right, so cyclic AMP, most common, these are some of the things we already talked about. So it can be activate and inhibit cyclic AMP synthesis. Now here we have the number of 1000 GPCRs. I gave you 800, depending on the source where you find these numbers. Even things like taste and smell are actually based on the GPCR mechanisms. In fact, now this is some signals that use cyclic AMP as second messengers. You can keep that for yourself in case you ever need to use this. So here, this just shows that arrestin, this is how arrestin works, right? So, so if this is our adrenergic receptor, you see it's a 7-transmembrane receptor. Now here on the cytoplasmic side of this, we have our trimeric G protein, and this one, this area here is the G alpha, the alpha subunit of the G protein. And you can see here, we don't have an X-ray structure of the adrenergic receptor, but we have a similar one of rhodopsin. Rhodopsin is also a variant of a G protein coupled receptor. So you can see here in rhodopsin, arrestin basically occupies the area where the G alpha would be. And arrestin will obviously block this area and not allow for the G alpha protein, G alpha, to come in and bind to it in preparation for the next cycle of signal transduction. So obviously GPCRs have been exploited not only by us, right, to make drugs. They have been exploited by various microorganisms to infect us, right? So for example, cholera toxin or pertussis toxin, they utilize GPCR signaling for their own needs, right? For their toxins. And so you can see here that there is in the alpha subunit of the G protein coupled receptor, or G protein, there's arginine that has an NH2 group. So it can react covalently here at the anomeric position of NAD+. And so pyridinium ion is a great leaving group. So the pyridinium ion leaves, right? And will form in a new bond and nitrogen-carbon bond. Now, can you tell me why this is a good leaving group? So this pyridinium ion, so we talked about halides, good leaving groups, tosylate, good leaving group. So why pyridinium ion would be a good leaving group? Just think about it. Is OH a good leaving group? Is this a good process? What do you think? From OChem 1, is this a good process? Is OH a good leaving group? No? No response? Okay, I'll give you a hint. It's a poor leaving group. How can you turn this into a good leaving group?
Acidic conditions.
Acidic conditions, that's right. So what would you do, and what the acidic conditions would do? Just keep talking. I'm just going to be drawing something.
Yeah, I would just make the OH form water.
Perfect. So we're going to be protonating this OH, correct?
Yes.
We're going to be protonating this OH, and then the nucleophile will come in and give us nucleophile can get rid of that plus water, right? OH2. So now using the same reasoning, why is this a good leaving group? Something about the charge. Here we added the charge and all of a sudden it became a good leaving group.
Well, right now it has a positive charge, so if it left, then it would become neutral.
That's right, and here it's neutral. That's right. So here it's neutral. So this pyridinium ion here, which is attached to the numeric position, is quite susceptible to a nucleophilic attack, correct? So cholera-toxin utilizes this very basic OChem 1 concept, right? Basically, turn this into a positive charge, make this into a good leaving group, and replace it with the carbon-nitrogen bond. Carbon-nitrogen bond. And so what happens once this ADP ribose, entire ADP ribose, is attached to the alpha subunit, it loses the ability to hydrolyze GTP to GDP, right? So it's no longer, this is not a GTPase anymore. Not a GTPase. If it's not a GTPase, what's the not the consequence? What's the consequence of that? So what's the consequence if the alpha subunit can no longer hydrolyze GTP? Yes. Well, if we shut off the signal by hydrolyzing GTP, and now we cannot hydrolyze GTP, then what? What's happening with the signal? It's constantly on, right? The signal is always on. So cannot be shut off. So this is how some microorganisms basically interfere with our signal transduction pathways and design toxins which do that. All right, so let's see how much time we have. We have about 5 minutes. Let's look over this slide very quickly. Very quickly. All right. So, okay. So this is a different type of different variant of GPCR, different variant GPCR. So this is not epinephrine. So this is we're gonna have a hormone that binds to a specific receptor. Okay, GPCR type receptor. And so let's see what happens here. So hormone will bind to the receptor. Now here's a trimeric heteroprotein. GDP leaves, GTP comes in, right? So in this particular case, it's going to be a different type of G alpha, right? This type of G alpha, or could be also a different cell type. This type of G alpha will bind phospholipase C, right? So this is an enzyme which breaks down the ester bonds with the lipids, right? And specifically, so by doing so it's a inositol based, so it's a phosphoinositol diphosphate. Okay, so it's the head group contains inositol. Remember I told you about inositol. Inositol is a pseudo sugar. So instead of having an oxygen in a ring, it has all 6 oxygens outside of the ring. So it's a hexahydroxy cyclohexane. Hexahydroxycyclohexane. So it's a very, so it's an important component of cell membranes, an important component of cell signaling. And so you can see you can phosphorylate any of these hydroxy groups. So phospholipase C basically cleaves off this head group from the phospholipid, which will then inositol triphosphate will travel through cytosol. Now it's highly soluble, right? It's water soluble. Not only it has a hydroxy group, it has 3 phosphates, and it binds to an ion channel, and it's a calcium ion channel. So it's a ligand gated ion channel. So once the IP3 binds to the channel, calcium leaves the endoplasmic reticulum. So one of the main functions of an endoplasmic reticulum is storage of calcium in a cell. So calcium is always kept at a very low concentration in cell. So mobilization of calcium, depending on the cell type, usually means that something needs to be done, right? So in resting condition, when nothing is happening outside, there's no invader, there's no signal to die, there's no signal to divide. So the concentration of calcium in a cell is low. But when something happens, this inositol triphosphate is released from the cell membrane, travels to the endoplasmic reticulum, opens the channel, and calcium pours into the cytosol and basically makes the cell, brings the cell to some kind of action. And so you can see here, so calcium binds to a protein kinase C, which is going to be anchored to the cell membrane through diacylglycerol. So by hydrolyzing this ester here, phospholipase C actually produces 2 second messengers. One of them is inositol triphosphate, and the other one is diacylglycerol. So both of these combined then activate protein kinase C. Protein kinase C, and that then results in cascades of phosphorylations, leading, depending on the cell type, leading to a variety of processes in the cell. So it's a different variation on the GPCR signaling. Let's see where we are. So it's 20 after 12. All right, so I think we're gonna stop here. So we have 2 lectures on this chapter. I think we've covered about half of the material. So we are on track. Do you guys have any questions about today's lecture? Any general questions?
No, thank you. Have a good holiday. Good break.
Okay, you too, guys.
Bye.
Stay safe, okay?
Happy Thanksgiving.
Yeah, same to you. All right, guys.